Transgenics is a form of genetic modification where a gene is taken from one species and implanted into the genome of another species. These are called transgenes. By using transgenes, a species becomes a transgenic organism. The target DNA is removed from the organism with the desired trait using restriction enzymes. The DNA is then transferred to the other species by inserting it in the desired spot on the DNA strand. The two genes are then bonded by using ligase. The transgene is then inserted into the zygote, where the newly born organism will be a transgenic organism. We have many examples of transgenic organisms today, including Bt cotton, a plant in which contains a gene from a soil bacteria that produces toxins to fight off pests. A glowfish, which has green fluorescent proteins extracted from a jellyfish, inserted into the genotype to create a fluorescent fish. Bt corn is a crop that has been genetically modified to have a bacteria genome within them called Bacillus thuringiensis. This gene helps the crops by producing a natural insecticide throughout their leaves that kills insects that consume it, protecting it from pests and preserving the crop. Crops like corn also have other problems when growing in a field. Weeds compete with the crops for water, nutrients, and sunlight. Farmers can use herbicides in an attempt to kill the weeds, but this could also result in damaging the soil and the crop itself. Instead, the corn can genetically be modified to resist the effects of herbicides and form a natural protection in their DNA.
Aside from using these methods on plants and animals, transgenes can also be used in humans. In earlier studies, mice have been used to model human diseases, and there are many potential and real ways transgenics can be used to enhance and/or create a substitute for key parts of the body. Dr. Curtis Connors proposed the idea of fusing lizard DNA with humans. He believed that with the right transgenes, humans could regrow limbs. This idea has not been pursued any further. The example that we have here of transgenics in humans is insulin, which is used to treat diabetes, of course. Now, natural insulin can be taken from the pancreas of cattle or pigs, but is limited in supply, and some people's bodies react poorly to it. As nowadays, most insulin is created using genetically modified bacteria that have had the human gene needed inserted inside of them. This process has many advantages over the natural one, as it can be made in very large amounts at once, so the limited supply problem is gone, and it is less likely to cause poor reactions from the body. To create genetically modified insulin, a DNA vector and specific enzymes are needed. A DNA vector takes pieces of DNA and inserts them into other cells. Viruses and plasmids can act as vectors. The enzymes that are required are called restriction enzymes, which cut DNA in targeted areas rather than just random places. And also required are ligase enzymes, which combine pieces of DNA together. The process of making genetically modified insulin is as follows. First, the gene that is wanted, which is the one for making insulin in this case, is cut out from a piece of human DNA using restriction enzymes. You then take an isolated plasmid, cut it at the point that you want to insert the new gene, and combine the two using ligase enzymes. The plasmid with the insulin gene is then inserted into a suitable bacterial cell. Then the transgenic bacterium reproduces, resulting in millions of identical bacteria that produce human insulin.
There are many benefits and drawbacks to transgenics, some of which include:
the first benefit to transgenics is crop productivity. Transgenics have given crops increased productivity by giving plant DNA that is already strong at fighting against diseases, and also allows plants to survive longer when there's dry seasons. The second benefit is that it protects crops. Things like corn and potatoes have been provided protection by transgenics by introducing a protein from soil bacteria called Bt. This protein has been used for many years as natural insecticide. The third benefit is that it can make food have better nutritional value. Over the years, scientists have used transgenics to make food better for you. In things like soybeans, they were able to make the beans higher in protein, and potatoes with more natural starch, which is healthier for people to eat. There are a couple of risks with using transgenics. Loss of biodiversity in the environment. Species in the environment that take time to grow will no longer be able to be the dominant species because scientists can make modified plants and animals at fast rates and make them better than the natural species that overall limits biodiversity. Genetically modified organisms include enhanced emergence of disease, pests or weeds, increased disease burden if the recipient organism is a pathogenic microorganism or a virus, increased weed or pest burden if the recipient organism is an invertebrate, and adverse effects on species, communities, or ecosystems. The fourth risk is the most notable risk to humans, are the potential development of allergens to genetically modified related crops.
Testing with transgenes can result in some ethical issues. The transgenic animals cause problems for some religions because they will restrict the food that the believers can eat. They do that because since they produce one animal that appears to be one species, they end up containing other elements of other species that are forbidden. There may be high mortality rates and other harmful effects on the animals used by the researchers to create the transgenic breeds. The disadvantages of transgenic organisms are that they include allergic reactions, emergence of superpests, and loss of biodiversity. Some people believe that some of the opponents of genetic engineering that transgenic crops might cross-pollinate with other related weeds to make a superweed, which will mean it is more difficult to control. Some people are worried that genetic engineering could possibly improve the plant's ability to escape and produce ecological imbalances and disasters.
Overall, transgenics is a useful way for scientists to overcome problems that can arise in humans and the essentials for our survival. In the future, we may find ways to avoid the risks and ethical obstacles, but for now, it remains only a talking point around the globe.